Ahora. Okay, hi everybody. Thanks for coming to our first webinar of the year. Today we have Gonzalo Parra from, he's a postdoctoral researcher at the Statistical Genomics System Genetics group at EMBL Heidelberg, and he's going to present us a great talk called Reconstructing Complex Lineage Trees from single-cell RNA-seq data using MERLoT. Gonzalo is from Argentina. He's going to tell more about the name of his development, MERLoT. Thank you, Gonzalo, for being here with us and for accept our invitation. And please go ahead with your talk.
Okay, thanks. Hi, everyone. Thanks, Jacid, for giving me this opportunity. So as Jacid said, I will talk about my work when I was a postdoc at the Max Planck Institute for Biophysical Chemistry in the group of Johannes Sodding. For those that are interested, this is my Twitter in case you want to have the discussion online, or you can write me later or whatever. So I'm currently a postdoc at the EMBL, but I will talk about the work I did before, the 2 years before when I was in Max Planck. So this is basically about these two papers that were published last year. So the first one is about the method MERLoT itself. And the second one, it's a tool we developed in order to simulate data sets to benchmark the tool. So I will start with this. So this is if we take a developmental process and we take the dimension of time, we can think of when a multicellular organism develops, we will start to have different cell types that emerge over time. And because of how they develop, we can think and put them into a structure of a tree, which means that certain cell types in time, after they perform different transcriptional programs, they will develop into multiple cell types that are ordered in this kind of tree. So the idea is, can we understand which are the transcriptional decisions that are made in order to get all of these cell types, and can we order these cells in this tree structure? So this is what we try to do. And a couple of years ago, thanks for the development of these two groups in parallel in Harvard, the one from Acosko and the one from Klein, they improved the techniques of microfluidics to give an enormous push to single-cell transcriptomics. So basically, they were able to improve the technique to somehow take a tissue, put it into different cells, and then put these cells into microparticles that will have everything you need to do enzymatic reactions like PCR and so on. And basically, after you do that, you can sequence little fragments of the transcripts and identify them as coming from specific transcripts in your cell. So at the end, you can have a developmental process like this one I am showing here, and you can take different samples at different time points, and for every sample, you can know how many transcripts you are having in that specific cell, in a specific time point, in a specific sample. So at the end, what we get, it's a matrix that has cells and genes, and we know at a certain extent, with errors, of course, how much of a gene is being transcribed in a specific cell. So this is the matrix with which I'm going to work and over which we develop our tools. So single-cell transcriptomics has different challenges. One of them, which is very common, is you need to define cell types. So the thing with single cell is that, let's suppose we have gene A and gene B, and we observe the relationship between these two genes. If we don't separate the different cell types together, we can enter of what we call the Simpson paradox, which is, if we take into account all of the cells at the same time, we can fakely say that we have a correlation which is negative and significative. But if we cluster these cells and we define we have the cell type I, or the blue one, and the cell type B, or the green one, we can see that the correlation actually is positive in each of the subgroups. And this is actually the real correlation that we have. So unless we define our cell types correctly, we can have artifactual correlations in our data. Another thing is that real time, it's not the correct coordinate to evaluate development in this kind of data sets. Because, for example, in the subplot in the left, we have the different cells that are taking different hours in the real time. And we can see that we have different cells from different cell types at a different time points. Because when we take into account tissues or different organisms, some cells, they are going faster than others in real time. But if we apply something that's called pseudotime, which is not the time at which we collect the sample, but we can think of a distance that is correlated with the transcriptional similarity from one cell to each other, if we consider some cell to be time 0, and then we calculate the distances in transcription information from one cell to each other, we can calculate this kind of pseudotime, which is what we are showing now in the x-axis, and order the cells according not to the real time, but to a developmental time. And then we can see this, of course, is a cartoon, is an example of cells progressively going from one progenitor type to a differentiated cell type. Another problem is that we are dealing with thousands of dimensions. Each gene is a dimension in our problem, and this is in the gene space. And the problem is that the relationships between the different genes are nonlinear. We have a lot of noise. Many of the genes are redundant. And of course, this is a problem because it's so multidimensional that it poses a problem in terms of efficiency for our algorithms. So with the use of MERLoT, our tool, we want to tackle some of these problems. Some of these problems are also solved by using other tools that are developed from before. Of course, we are not reinventing everything here. And I will talk a little bit. So what is MERLoT? So this is kind of a guide of what we can do with our pipeline. So if we have our process that we have a group of progenitors and 2 mature cell types, we can sample this process at different time points, and we get our matrix of cells times genes. This matrix is high dimensional. It can have 20,000, 30,000 dimensions. So somehow we want to first lower the dimensionality of our problem. And we do this by applying different techniques, but basically it's a PCA or a TSNE or a UMAP. Some of these things are very known in the field. So basically you go from a very, very high dimensional space to another dimensional space in which a few of these dimensions, they capture most of the information, and we can keep 1, 2, 3, 4 dimensions and work in there. So once we have our dimension with our cells that are embedded in this low dimensional space, we can start to work. Basically what we do is we play with the distances. Now we apply Euclidean distances between the cells, and we can create this kind of what we call scaffold tree, which is in this developmental process that we have our progenitors and our cells in the mature states. We want to know which is the structure of our tree. Of course, it's a very simple example. Again, it's just a cartoon for us to understand. But what we want to do in this step is to localize where the endpoints of our tree are and where the branch points are located, and also how the endpoints and the branch points, we can also call them wave points in different papers, how they are connected to each other. So do we have a normal connectivity? Do we have a star? Do we have a circle? Which is the connectivity that we have? This we do with some techniques that I will explain later. So once we have this scaffold tree, we apply something called an elastic principal curve. With this principal curve, what we want to do is to solve. If you see the scaffold tree is kind of ziggy, it has kind of this is not uniformly distributed across the density of the cells. So with the elastic principal curve, what we do is we soften, we smooth the structure of our scaffold tree, and then we put these artifactual, non-real cells, these black spheres in between, and we put them in such a way that they interpolate correctly the density of the cells. So now these are not real cells anymore. These black points, they are fake cells in the low dimensional space, but they are useful for something I will explain later. So once we have this smooth tree, what we can do also is to apply pseudotime. And for this, we say, okay, our endpoint located close to the progenitors is our time 0. And then we are going to assign different time values, pseudotime values for the other cells based on how far along the graph, they are located in the tree structure we just constructed. So I said that these black dots, they are not real cells anymore, but they are in a space which is our low dimensional space, which is calculated from the high dimensional gene expression space that we had before. So we can think of saying, okay, we can consider these fake cells as pseudo cells, and they are located in a region of the space which has somehow the information from the genetic space in which they were before. So what if we take each of these black points and we say, how about taking all the cells that are around them and asking for every gene, every cell, which is the gene expression value for every gene from which they come in the high dimensional space. And then we take, let's suppose a cell has 20 neighbors, 20 real cell neighbors. So let's say we have gene 1, and we take the value of the gene expression for this gene 1 for every of the 20 cells around, and we calculate an average gene expression value. And this is what we have here in the step 4. Let's suppose this is gene expression value in the X, and then we have our cells. Every row is a cell here, and every column is a gene. So we take the average expression value from all of the cells around a black point, and we put this value there. So high, it's darker, low expression is white or yellow. So we see that this matrix, it's kind of noisy, and there are some rows that are completely red. These red rows are those black points for which there's no neighbor around, because every cell can be assigned to only one black point. So some black points, they have no neighbors at all. So these rows that are completely red are those for which the expression of every gene is 0, because they have no information. But we know the exact location where these black dots are. We know how they are connected. We know that one black point has a direct connection to one before and one after. And we have all this spatial information and time information. So we can apply also our imputation, our principal curves, in such a way that we take the information and the connectivity from every cell, and we just smooth every coordinate in the high dimensional space, and we go quickly from step 4 to step 5, in which you have a smooth new imputed gene expression value for all of our black dots here. So you can see that more or less the information is the same, but it's softer, it's smooth in the last step. So after this, we can take again our pseudotime that we got in step 3, and we can now check the expression value for all these black dots, which are the continuous lines here. And for every gene, the continuous lines are the imputed values, which are the values that we have in these matrices in step 4 and 5. And then we can also put the real values from the cells that were the neighbors from which these average vectors were calculated. And in these plots are the gray points that are around the imputed values. So we can think of these imputed values as the ideal expression values that we will have for our cells if there were no noise, both technical and biological. And then the cells are just scattered around these ideal values. So let me now go ahead and I will show how we apply MERLoT in a real study. So this study, it's not single cell, specifically real single cell RNA-seq data, but real-time qT-PCR. So why we do this is because this is a high quality data set. It's about the development of blastocyst from zygote cells in mouse, and we have only 48 genes, which are all important for the developmental process. And so it's a low dimension, it's a low number of cells that we can manually check to understand if we are doing things okay or not. So let's continue. So we have a total of 428 cells, 48 genes, as I said, and these are sampled from different time points along this developmental process. So the first thing that we have to do, we have our matrix with cells and genes, so we have to reduce the dimensionality of our problem. So For reducing dimensionality, we have different techniques. So the idea is to go from the full gene expression space to something which is with less dimensions, but also where things like the Euclidean distance make sense. And there are many ways of doing this. This is not the focus of my talk, but there are different methods. They all can relate at some extent to PCA, which is principal component analysis, but we have different ICA, LLA, TSNE, diffusion maps, DDR3, there are many. So the idea is that we don't care about it. We give this responsibility to the user, so the user can just transform the data and calculate these other spaces, and then we keep the most informative dimensions, let's say the first 2 or 3, and then we give these dimensions to MERLoT as input. So in that case, we are very flexible. We accept any type of coordinates that the users can give us. So for the data that I just described, this is what we get. So axis 1 is a diffusion map. So the first axis is the diffusion component 1, and the second one is a combination of diffusion component 2 and 3. These are three-dimensional plot, but for improving the visualization of the data set, I just flapped everything into the plane and just represent it as a two-dimensional plot. So we have the different cells with different colors. So the 2 cell state is in red, 4 is kind of this mustard, and then you go to the 64 cell state in blue. And you can see that somehow the colors, they are grouped according to the state in which they are located. And then you have 3 clouds of blue points, which represent the 3 embryonic lineages that you have at that state of the blastocyst. So the idea is, can we say something about this process with MERLoT? So the first thing is that we have to calculate the scaffold tree. The scaffold tree, also very briefly, we apply the Dijkstra's algorithm, which it's about trying to find the shortest pathway across many connected cities. It's also called the Salesman's algorithm. And we, instead of cities, we have cells, and instead of roads, we have Euclidean square distances. So we calculate the shortest pathway around all of them, and we try to see how is it to go from one to each other. And then we combine this with the neighbor joining algorithm, which is also a very famous algorithm for phylogenetics, which is to understand how the structure of a cladogram is built. So we combine these 2 things to get our scaffold tree. So this is what we get. So we have in total four endpoints, 1 in the red and 3 in the blue clouds, and then we have 2 branch points, 1 in the green cloud there, and another one in the yellow one. So we go from the zygote, the 2 cell state, we go up to the 16 cells state, and then we split into 1, which is the one of the lineages for the embryon, and then we go to the innermost cell, to the cyan cells, and then they split into 2 other lineages. The problem with this scaffold tree is that it's very robust. It works very well and fast, but it's sparse. As I said, it's SIGI. Not all, it doesn't interpolate the density of the cells very well. Some regions are empty, so we have more cells there than in other space. So there's a bias of information towards certain regions of our plot. So this is nice, but not perfect. So how do we solve this? With our principal elastic trees, this is the citation. And with this, we apply our elastic tree, and we have our cells. In this example, we apply 100 nodes in our cell, and these are these black pots that we put here. And as you can see now we don't have real cells anymore, but these fake cells, they are distributed nicely across the space and in a very homogeneous way. Once we do this, we can assign every cell to its closest tree node, and we define these as clusters, and based on markers from the original publication, we can assign these different cell types. So we have the zygote, we have the trophoectoderm, is the violet one, we have the internal cell mass there in orange, then we have the pseudoendoderm in blue, and the epiblast in red. So far so good. What are these interesting for? So this was an example, but we have different data sets. So for example, in the first panel, you have a blood sample, which is a single bifurcated topology. The second one is the one that I just showed, which is double bifurcation data set, and then we have much more complex structures than the one in the panel C and F, which is also block development, but from a different time point, so we have more cell types emerging. And the second row is just the reconstruction of the tree that MERLoT does with the assignment of each group of cells to the nodes. Also, we can go to more complex stuff. For example, we can have some trees are very complicated, so it's not possible to visualize them in 2 or 3 dimensions. For example, this data set here, it was calculated in 10 dimensions in our diffusion in a diffusion map, and it has different cell types there. And what we can see is that the panel A, it's what you get when you apply a reconstruction using monocle, which is one of the algorithms that was present there before we published MERLoT, and panel B is what we get when we apply MERLoT, and what we show here is that we make a better reconstruction of our tree based on the cell types, because, for example, you can see that the blue cells are better clustered here because they, in panel B, they come from this internal node, and then they go up, they separate in this individual branch, whereas in the panel A, they are mixed between the 2 branches. So basically, what you see, the blue cells that are in the upper branch, they are badly assigned. So far, we showed that we can reconstruct the structure of our trees, but what is it useful for? Because at the end, what we want to do is to study gene expression. So far, we have a tree structure which looks nice, but it's not useful for anything. So the idea is that we can apply now we show why the last part of our pipeline is useful. So as we say, we have pseudotime. Pseudotime, it's just the distance along the tree from every cell compared to a time 0, which is our progenitors. And so this is the time, the pseudotime we have for the toy example dataset I showed first. So the first panel has a pseudotime for the nodes, and then each cell will take the pseudotime value from its closest node, which is what I'm showing in the panel in the lower part. So once we do this, we can get, for example, these are 4 genes that are important for development. They are markers for the specific cell types, and we can plot them as a function of pseudotime, and they are colored by the cluster to which they assigned. So you can see some patterns here. Somehow the colors are sometimes separated, but can we improve this? So the idea is that we have our pseudo cells, as I explained. We take for every K node in the tree, we take the cells around and we calculate our average values, which is what I'm showing in the matrix in the top part of the plot. As I mentioned also before, the red rows are nodes for which we have no information, no cells around, like for example K4. So we give a vector of 0 expression. Once we apply our principal curves algorithm, we get from the upper panel to the lower panel, and then we have our smooth values. And what we can do is, if we take these imputed values and we superpose this in the plots that we had before, we can have something like this. So now we can clearly see the structure of our developmental process on top of the expression of the genes as a function of pseudotime, which is something super useful for understanding the processes. And you can imagine that some processes are much more complicated than only having 4 cell types and 2 bifurcations. So what else can we do? So we reconstructed the structure of our tree, we calculated pseudotime, we calculated the ideal imputed gene expression values for the nodes in our tree, and we can superpose it in our pseudotime gene expression plots. So is this useful for something? So we have this data set from our collaborators in the Barbara Treutlen's group. So this is a group of iPS cells, these induced stem cells, and they are somehow forced to develop from neurons from fibroblasts to neurons. And the thing is, so they start from the MEF cell type, and they are trying to force these cells to go to the neuron's lineage. But this process is not 100% efficient, because some of the cells, they are very rebels, and they say, I don't want to become a neuron, I will go and become a myocyte. And this happens more or less in 80% of the cases. So the idea is, can we know why this happened? Can we check which are the different genes that are expressed in each of these cell lineages? So this is the diffusion map. For this data set, we have 390 cells and 290 highly variable genes. We are not using all of the genes, but only the highly variable ones. And we have this single bifurcation data set here. And this is the tree that we reconstructed with MERLoT. So we apply all of the things that I described, and we can have our genes and our imputed values, and we can observe how every gene behaves across the tree. So for example, AP3B2 is a marker for neurons, and we can see that it's highly expressed in neurons and not in the others. And the same happens to S100A6, which is a marker for the myocytes. So how can we do? If we calculate, you remember the Simpson's paradox, in which I was talking about correlation between genes. If we observe the correlation values for two genes, and these genes are highly correlated, we will see a strong signal, but this signal can be influenced by how much noise we have. If you have these two genes here, you see that the imputed values, they go in the solid lines, but then the real values from the cells, they go all over the place. We have a lot of zeros in the bottom part of the plot, and then they are scattered all over. So we can try to calculate pairwise all the correlations we have for our 290 genes, but we can do both. We can calculate our correlations based on the real values from the cells, or we can do it using the imputed values that we just calculated with MERLoT. And we can use this, for example, and ask, are they cool to calculate gene regulatory networks, for example? Because we could eventually calculate correlations between every pair of genes and think that every gene is a node in our graph, and the correlation value is the strength of the connection between these two nodes. And then we can apply a physical model to calculate, okay, things that are really correlated, they are close in space, and things that are not correlated, they are far in space. And after certain cutoff, we can say these two genes are not connected at all. So for example, we can calculate something very, very simple, like the Pearson's correlation for every pair of genes. And we can say, okay, if we have a Pearson coefficient higher than 0.8, we consider these two genes to be connected. And then we apply these layouts for checking which is the structure of our network. So we did this for this data set, and we have something like this. So here, these are the 290 genes that we have, and these are the labels, and we just clustered this graph with different colors, and we try to understand now if this makes sense. So we took the genes that are highly expressed in every group of cells in our tree. For example, which are the genes that are highly expressed in the branch, in the red branch, and we did both things that are upregulated or downregulated significatively, and this is what we get. So if you check what are the genes that are highly expressed in the red branch, is what we do now in everything. So in panel B, sorry, we have the fibroblasts, and things that are highly regulated are things that are in red, things that are downregulated are things that are in blue, things that are not either high or downregulated are in black. And we can see, for example, that for fibroblasts, we have this red concentration of genes that are all together, and they are related to mitosis and proliferation, for example. In neurons, you have this other, in panel C, you have this other group of genes that are highly expressed, and they are all related, for example, to processes related to synapses. And if you have the myocytes, for example, you have all the myogenic genes that are highly expressed there, and as the other side, all the ones that are, if you compare neurons and myocytes, you can see that they have complementary transcriptional programs, because the synaptic genes are not expressed at all in myocytes and so on. So with this, it's just a small example on how we can use MERLoT to do multiple tasks up to maybe the reconstruction of gene regulatory networks. And of course, this is a single bifurcation process, which is not very complicated, but it was just to show the potential utility of our tool. I must say also that this is not a real gene regulatory network, because we don't know causality here. We don't know who is regulating who. When we have two genes that are connected, we don't know which is the causality, which is a correlation network. Basically, it's not a gene regulatory network, which is a good comment, I should say, because otherwise they would think that I did things that I did not. But yeah, so to conclude, we developed MERLoT, which is it can robustly reconstruct the lineage tree topologies. We did a benchmark, which I can comment later if I have time, if someone is interested. We simulated data sets with up to 12 bifurcations, and we show how do we perform compared to other methods, and we were very good at doing this. Also, we can reconstruct the gene expression time courses by embedding our tree structure into the full gene expression space, and we can understand how our genes behave in our developmental process as a function of pseudotime in different cell types that we have. And then we can use the tree structure to easily identify genes that are differentially expressed in different parts of our tree, which can serve as markers. And finally, we did a small example of how we can use these imputed values to maybe start to reconstruct gene regulatory networks if we add somehow the causality issue into play. So this, just I want to acknowledge the group of Johannes Sodding, my PI when I was in Göttingen. Also, these are 5 students that were helping me along these 2 years that I was there, and they worked very hard with me, and also the group of Barbara Treutlen, that they helped us to understand the biology behind all of this single cell, because when I started, we had no idea. And of course, to all of you for attending this webinar. So thank you very much.
Thank you, Gonzalo, for this great talk. Can you hear me?
Yes, now I can hear you.
Yeah. Can you hear me?
Yes, I hear you.
Okay, thank you. Okay, thank you, Gonzalo. And now we have time for questions. If any questions, please write on the chat that you have available. Okay, questions. Okay, I have one question from my ignorance in this field, because I'm not really working on single cell RNA-seq. So in terms of reconstruct complex lineage trees, we can say that in cancer, for example, in metastasis process, we have different kind of lineage trees from different kind of cells. How you can apply MERLoT in order to reconstruct the metastasis process and the process of differentiation in different types of cancers related with different kind of tissues using single cell RNA-seq data?
Yeah, so this is something we are trying to do. Actually, the cancer it's complicated, because you have your healthy patients. So basically, you can have healthy patients and compare them to samples from patients that develop the cancer or the disease, any disease, because you can also do it, for example, with Parkinson or whatever other genetic disease, and try to understand which are the genetic differences among the cells that come from healthy tissue and the tissue that has a disease, basically the tumor samples. So, I mean, we can do this, but then we need to redefine the concept of pseudotime. So can we say that cancer progression happens in a time-wise way? So we can think that we have normal tissue, healthy tissue, and that the cells, they accumulate mutations and they start to apply a differential transcriptional program that takes them from the normal phenotype to the diseased cancer phenotype. And we can think that this happens in a progressive way, in such a way that we can order the things in a pseudotime way, right? So we have a pseudo-cancer time point now. If this happens, then we can apply MERLoT in this. Like we will have, for example, samples from our patients that then they maybe have a tumor, they have surgery, so we have samples from this tumor, and let's suppose they have surgery and they can have after a relapse, and they have surgery again and they have maybe another relapse. So we have samples, actually this is a real case that we have now, so we have samples from healthy patients and the tumor and relapses, but we are not sure that the relapse it's really different from the primary tumor, or if you have metastasis, for example, because cancer happens also in a clonal way. So is it a single clone that gives rise to the metastasis, or are there multiple clones that coexist? Do these changes occur in a progressive way in the lineage? So cancer is kind of complicated, but yeah, of course, I mean, we are trying to do this, and we are trying to discover if, for example, we can find which are the genes that get perturbed across the cancer process. And for example, if we can reconstruct our gene regulatory networks, the hypothesis would be, can we detect which is the gene that is responsible for the phenotype change? And is it a gene that in our network is related to many other genes that are all related to each other? And can we maybe find a treatment attacking these target genes that we identify? So this would be more or less the idea, but yeah, I mean, we are trying. I don't know if this is possible at all.
The other thing is that using single cell RNA-seq, you have less noise than using RNA-seq, the normal one.
Yeah,
right?
Yeah, we have much more noise in single cell.
Much more.
We have much more noise in single cell, and also we have a problem, which is a dropout problem, which is for many of the genes, we have no detection. So basically in our matrix, we have a lot of zeros.
Okay, got it.
That's why you need to sequence a lot of cells, because the idea is that many of the genes you will not detect because of a technical limitation. But if you do this thousands and thousands of cells, you will eventually catch the gene in some of them. So let's say for many of the cells you have zeros, but for some of the cells you will have real expression data. Okay. And this you do by having thousands of cells. So the single cell is cool, but you need much more cells because you have a lot of sparsity. And because you need a lot of cells, you have to play with the algorithms, because then the algorithms become very inefficient at dealing with so many cells in your data set.
Okay, thank you, Gonzalo. Okay, last opportunity for questions. Okay, if we don't have questions, maybe you can send an email to Gonzalo to ask for doubts or something else. Okay, Gonzalo, thank you again, and
thank you.
See you. We hope to have you here next time.
Okay, thanks a lot for the invitation.
Okay,
bye-bye.
Bye, Gonzalo.